It's actually very good. They built it as an entry-level machine, and various companies stopped selling it because once people look at the specs, they said, well, why would I go for the other one with a laser that's going to cost me a fortune? I'm going to go with a LED where if it breaks, it's much easier, much cheaper to fix. So this one, again, has beads with a specific spectral address. And forgive me, someone spoke about this yesterday. I can't remember your name, but I do acknowledge that you were talking about this. So you have beads with a specific spectral address. As I said, it reads up to 50. So it means you can look at up to 50 analytes. This is just showing you the region that the mag pixel read. The LX200, which is the other model, will read up to 100, and the FlexMap 3D would take up to 500. It's very simple, as I said. You've got a CCD camera and two LEDs, and a magnet, which you suck up your beads with this fluidic system. You adhere your beads to the magnet. You take a quick picture using both these, using the CCD camera and your two LEDs, and that's it. You go to the next bead. So it takes you about an hour to look at to read 96 wells. And in each well, you can have up to 50 analytes. Again, much like the microarray, it goes way beyond looking at cytokines or antibodies. You can look at just about anything. Groups in Uganda are looking at drug resistance markers or SNPs within the propeller, the Kelch protein. So you can do, again, imagination is your only restriction with these systems. As I said, optimization is really key with these systems. Because although they can give you a lot of information, trying to get it to a point where you can trust all the data that comes out is key. And I thought it was fitting to show an example of RH5. And with this one, what you can see is the fact that we use a number of different serological controls. And you can see there's a split in those controls that actually react to RH5 and those that react to RH5 and those that don't. So when you're working with the bead-based system, you need to be able to optimize your coupling efficiency on your beads and the coupling concentration onto your beads. Because without that, you can't control the data that comes out. You can overcouple, and that can lead to aberrant data, or you can undercouple when you're not sensitive enough. Again, this is another marker that we identified using the Feldman array when we're trying to identify markers of short-term exposure. And you can see there's a greater response in terms of the way people respond to this. I mentioned this idea of markers of short-term exposure in a paper we published last year. What we're really after, when we're looking in these sort of serological surveys, we really understand in regions where we're, and I don't want to say this because someone put E up and they didn't even say elimination eradication, so I'm a bit nervous about this. But when you're looking at regions where you're investigating elimination or eradication, you want to be able to measure zero. You want to get as low as you can. And so your markers have to be telling you when someone was last infected, and hopefully you want that resolution to be a year or less. And so we've identified a number of markers for which when someone's infected, we can tell when they were last infected. We can see that resolution drop over 3 to 6 months. But I'm not going to show you that data. We still need to validate it. So what about sensitivity? Your LOD, almost done. Your LOD is much greater. So looking at the microarray data, you can see with your ELISA versus your microarray, if you put a line across here, anything beyond that line is being picked up by your microarray, that isn't being picked up by ELISA. And actually, when you think about it, with your ELISA, you're looking at OD cutoffs of around 1.5 to 0.2. And so anything beyond that is being picked up in microarray, that isn't being picked up by ELISA. So your LOD is much greater. When you look at the magpix, you can see the red line is showing you your ELISA data. You can see you saturating with your ELISA. But when you look at the blue line, which is still straight, your LOQ, or your limit of quantification, and your limit of detection is much greater. There's much greater range of what you can pick up. So with systems like this, you don't really want to look at this in a binary manner. You want to look at this, you want to appreciate what the range of reactivity is. So just to summarize, using our system, we've developed up to about 230 malarial antigens. This is increasing all the time. We've got candidates for falciparum, for Vivax, and for Nolzi. We've also got a number of non-malarial antigens because we want to put a number of non-malarial antigens because we answer questions about immunity. I'm about to get kicked off, so let's speed up. And so we got further candidates that we want to include. So these are really just summarizing the fact that the microarray can allow you to screen large numbers of candidates and gives you a huge number of data points. The data you can get can help you monitor changes in serological prevalence, changes in transmission. And this one's basically showing you data from Vanuatu, where as you go from 1998 to 2008, you see the change in antibody prevalence as you go over the years. And this is a region where they're able to eradicate malaria, but then it's an island, much easier to affect. And this you've seen. And so what we have is a system where we can screen large numbers of analytes, and we can triage down to different platforms depending on what we want to do. And I'd say thank you, didn't do this work on my own, and I'm done.
Thank you very much, Kevin. This is a gentleman who said to us that he's a shy person, but I don't see that in him at all. Any questions?
It's really excellent talk. With your protein purification, did you clip off the affinity tag before, or does it make any difference?
No, we didn't, because so we express using the GST system. And we always get questions about whether the GST is going to give us any background reactivity. But the way we optimize the ELISA, the Luminex, and the microarray, we don't have any aberrant reactivity with GST. We even acquired samples from HTD of people who have contracted schisto, and of course the immunological reactivity with schisto is very different, but even screening those, we don't pick anything up. We look at those individuals with the purified GST, we still don't see anything. So we correct for that. We don't cleave the tag off, because sometimes when you cleave the tag off, you lose solubility. So the GST, MBP, thyroidoxine, all these kind of candidates actually act as a kind of solubility factor. So when you remove that solubility factor, you can actually lose the ability to stay soluble that they provide. And so the idea is to screen them and correct for the background. And then when we go further, there were other things I was going to say, but we're out of time. But if we were to go further, there's other questions we want to ask, and that will involve trying to re-express these with his tags or in other systems.
Okay, Kevin, thank you very much for a very nice presentation. I have a question about the seroconversion rates.
Yeah.
So that is an annual seroconversion rate that is being estimated based on the ages of the individuals that you sample from. I have an issue with when you have to, or when you do this work in an area of seasonal transmission, when within the year you are having changes.
Yes.
This is not able to detect that, I guess. Is it possible to do this maybe on a monthly basis instead of a yearly basis?
It would depend on the data points that you have. So I'm not sure. I'm not sure about that one. I'll have to look that up and get back to you. But we have run. I mean, the thing is, you're right. When we run the seroconversion curves, we can't run it in every setting. Some settings it works, and some we can't get the curves to fit. So in those instances, we have to try and think about other ways we try and analyze the data, but it doesn't fit every situation.
Yeah, that was a really beautiful talk. Thank you. The technology is outstanding. The question that I have is, if we ask a simple question about clinical immunity to malaria, for which we still have no definition, how do you decide what to spot down, what your critical threshold as a discerning factor would be? I mean, you can always put more in, and we think more information is good, but how do you know magnitude of scale in order to threshold it? I mean, do you use 1,000? Do you use 10,000? You see what I'm saying in terms of what you're spotting down?
Yeah.
Isn't in the end it really making a model that's the best discriminator with a lot of data, reducing it down into almost like a PCA type of analysis?
Yep.
Have
Absolutely.
you thought about doing that?
Absolutely. No, for certain. So it's a very good point. So one of my collaborators, did I put him on there? Yes, I did. One of my collaborators is Brian Greenhouse, and we work very closely with Brian. And so what we have done is once we get this data, we kind of fit it into models. So with some of the serology data, we did that with some of the candidates. In terms of magnitude, that's a good question, because some of the candidates that people have been pushing as vaccine candidates, the prevalence and the intensity of reactivity is quite low. So what we're hoping to do is we're not being guided by the intensity of the response at this stage. We just want to see if we can interrogate data and put them into groups. If we get antigens which are telling us the same thing over time, we can narrow that down. We can look at things like, for example, Shizon egress antigen 1, RH5, things like that, where there's a lot of supporting data. We want to see how we can look at that in an epi context. And so, yes, models and everything to put those into context.
Thanks a lot, Kevin.
Thank you. Thank you very much.
So the next section is for the fellows. The next speaker is Christiana Ofori Owana.
Good afternoon. My name is Christiana, and the title of my work is High-Resolution Melton Analysis of Drug-Resistant Alleles and Multiplicity of Infection in Plasmodium falciparum Reservoir in northern Ghana. I was supervised by Dr. Anita Gansa and Dr. Gordon Awandari. So malaria continues to be a problem globally, and with half of the world's population at risk, malaria control is key to reducing the disease burden where it is no longer a health problem. Malaria elimination is a state where interventions interrupt transmission in a population, and this requires continuous intervention measures and constant surveillance. So usually malaria control is geared towards clinical cases, but in regards to malaria elimination, targeting all malaria infections that contribute to transmission of the infection is key, regardless of the symptomatic status and parasite density. Now, one of the manifestations of malaria is asymptomatic malaria, and the frequency of these manifestations is higher than originally assumed in malaria endemic areas. So this is defined as a case where an individual has paresthemia at any density, however, in the absence of fever or acute symptoms. So this happens when an individual is repeatedly exposed to the infections and thus develops a state of immunity, as well as a state of protection from clinical symptoms. So these chronic infections are known to constitute the reservoir of infection and is involved in fueling continuous malaria transmission. However, as elimination is being attained, already the paresthemia of these infections are at low levels. So as you're attaining for malaria elimination, they would definitely decrease. And so this requires more sensitive detection tools. So my aim was to adapt and utilize the high-resolution Melton method to describe drug-resistant haplotypes and multiplicity of infection in the Plasmodium falciparum Reservoir in 2 communities in the Bongo district of Ghana. So my objectives was to adapt the method in asymptomatic cases, to assess the frequency of drug-resistant alleles, to determine the multiplicity or complexity of infection circulating within this reservoir, and as well to assess the correlation between drug resistance and complexity of infection. Oops. So for my study site, my study site is Bongo, which is one of the 9 districts in the Upper East region of Ghana. And this site has been selected for malaria elimination studies. So 2 communities were selected. There's Via and then there's Soin up there. So from both of these communities, 2 villages were selected. So from there, it was Via and Guari, and then from Soin is Soin Boko and Soin San BC. So for my laboratory analysis, 200 archive samples were selected, and DNA extraction was done using a QIAGEN kit. And for both drug resistance and complexity of infection, the high-resolution Melton method was used. This method has been developed by Daniel et al., and it involves the use of is a post-PCR method, which involves a high-class saturated DNA binding dye, which fully saturates your DNA. So after amplification, once you denature your DNA, a Melton curve is a Melton curve is shown. And for this assay as well, a lunar probe was involved, and this probe is unique in the sense that it uses, it is unlabeled, and it's not fluorescently tagged, and is complementary to your wild type allele. So for the mutant allele, there's a mismatch, which causes a shift in the Melton curve, which allows you to easily see between the wild type and the mutant. So for drug resistance, these genes were studied. There's the dihydrofolate reductase gene, where a mutation at that codon infers resistance to pyrimethamine. And then there's also the multi-drug resistance gene, where a mutation at codon 86 also infers resistance to chloroquine and mefloquine. And then I also looked at the chloroquine resistance transporter gene, where also infers resistance to chloroquine at that codon. For the complexity of infection, another method developed by Daniels et al. Was used, a molecular barcode assay, and what was done was 24 SNPs were selected, and you can see they're distributed across the genome. So these are neutral, unlinked, or common, and as I mentioned before, they're distributed across the genome and gives the true identity of the parasite. So for my work, it was actually 10 that were selected that were informative to our population of interest. So for the results from the figure here, you see when you're looking at parastemia in the Bongo district, you see within the age group there's a high frequency of parastemia as expected, and this was significant when being compared to each of the other older groups. And as well for when looking at the community level between VIA and soins, you see high levels of parastemia as well. And this could be explained, sorry, I didn't mention it before, but VIA is close to an irrigation dam. So in this case, there's more breeding sites. So the people in that area will probably be more exposed to infections, and they'll probably develop a state of immunity. So therefore, you see those low parastemia levels. And then for the frequency of the drug-resistant alleles that we looked at, from this figure here, the blue bars represent the wild-type allele, and the red represent the mutant allele. And the first two are from each of for the villages that were selected from VIA, and then the other two from soins. So for both CRT, for CRT and MDR, you see high levels of the wild-type allele across the villages. And this could be explained by the removal of chloroquine from Ghana, and several studies have shown the re-emergence of the wild-type allele. However, you see the opposite with the DHFR, where you see definitely very high levels of the mutant allele. And this could also be due to the use of sulfadoxine pyrimethamine for intermittent preventative treatment in pregnancy. So for the prevalence of the CRT and MDR haplotypes as well in the Bongo district, when you look at the PFCRT for this genotype, you see a high level frequency level. Again, this is, like I mentioned before, due to the withdrawal of chloroquine from the system. And then for the MDR, these are the haplotypes we noticed in all of our samples. There were high levels of the NF and high levels as well of the NFD. And this is also similar to other studies where previous studies also done in Ghana by Dua et al. Showed these similar high levels of the NFD. And as well, Thompson et al. Reported in Mozambique that for the CV, MNK, and the NFDs, these were selected for after the replacement from chloroquine to the ACT combination therapy. Now, this also is a pictorial view that depicts the diversity from our molecular barcode assay. So these are some samples from VA and from SWE, and these are the different, as I mentioned, the 10 SNPs that we looked at. So as you could see from their diagram, there's definitely high diversity. And for these ones which are seen in these colored boxes for VIA, there was only one barcode that was similar in 2 samples, and that was the same as well in SWE. And there was also one where there was one in our isolates. There was a sample where it was identical both in VIA and in SWE. So when comparing the mean complexity of infections by age groups, you could see from this diagram as well, high levels across all age groups. However, the ones that were significant was between 1 to 5 years and 11 to 20. And again, this can be explained by immunity. As well, we compared the complexity of infection to the drug-resistant alleles, and there was only a significance when looking at the CRT codon 76. So as well, you could see there are higher levels of the wild-type allele as opposed to the mutant allele. And this, again, is likely due to an individual when they are harboring more than one clone. As I mentioned before, with the reemergence of the wild-type allele, there's competition within these clones in the absence of drug pressure. So due to the fitness cause of this mutant allele, you definitely see a higher levels of the wild-type allele. So for my summary, we saw in the study a re-expansion of chloroquine-sensitive parasites in Bongo, a near fixation of the DHFR mutation, and also a high diverse parasite population within Bongo, and a trend of a higher complexity of infection in VIA, which is the irrigated area. There was more complex infections in older individuals as opposed to children, and as well a higher mean of the COI, specifically for the CRT allele. So for recommendations, it would be good to continue this study again and compare within seasons, and as well, due to the high frequency of mutant alleles with the DHFR, to consider a different alternative to the SP treatment. So I'd like to acknowledge my supervisors, all these people, and WACPIT as well.
Thank you very much, Christina. Quick question. High resolution Melton analysis of track of alleles.
Yeah.
That phrase, can you break it down for me?
Okay. So as I mentioned before, it's a bit of a refinement to the Melton curve analysis. So the unique thing is the use of sort of novel dyes, which fully saturates your DNA. So it's included in your PCR reaction. So after you amplify your DNA and you denature your DNA, then there's a decrease in fluorescence. And from that, you'll be able to depict the differences.
All right, thanks.
Yeah, something that I've been trying to understand for a long time, maybe you can help me. So in the high transmission region, right, as you went into older individuals, and this is not with the resistant alleles, but you had a higher complexity of infection, fair enough? Mm-hmm. Is by de facto the rationale that those individuals have not developed immunity early on, they're seeing different complex infections is why they're clinically sick? Because I didn't see the information. I'm assuming since they have parasitemia, they were clinically ill, but I don't know that. That's a question. Were they clinically ill?
When these samples were collected at the point in time, they were not clinically ill. So these were, the exclusion criteria is if you had parasthemia and a fever as well, then you're excluded from the study. So these were not clinical.
Okay, Christina, thanks a lot. Okay, so after this speaker, I will hand over the mic to my co-pilot, and then he, sends us safely to the airport. So it's Sandra King, Sandra A. King, the next speaker.
Good afternoon. My name is Sandra Adelaide King, and I'm presenting on evaluation of biological response of insecticide susceptible Anopheles gambiae bred in field water from an agricultural area. Malaria is the #1 vector-borne disease where about 219 million people are infected annually, which results in about 600,000 dead. Malaria in sub-Saharan Africa is also known to be transmitted by the Anopheles gambiae mosquito. In sub-Saharan Africa, urbanization is said to be a problem which contributes to malaria. It's commonly assumed that urbanization leads to decrease in malaria prevalence due to factors such as reduced biting rates, due to high human ratio to the mosquitoes, and also reduced fewer breeding sites, and humans also trying to live in protective housing. However, areas with unplanned urbanization that are typically associated with poor sanitation, poor education, may not experience such marked decrease in malaria transmission. Some of the activities that have contributed to this is small-scale urban agriculture, which usually creates breeding sites for the mosquitoes to breed in. So for the rationale of my study, Anopheles gambiae is usually known to breed in clean water. However, current evidence suggests an adaptation to breeding in polluted breeding sites, which leads to an increase in breeding sites in urban areas, also leading to an increase in malaria transmission, as a result becoming a challenge for malaria vector control. Furthermore, insecticide residues and xenobiotic could lead to an increase in detoxification enzyme, hence the mosquito's ability to adapt to these breeding sites. And unpublished data also has suggested that an adaptation of Anopheles gambiae to polluted water due to the presence of due to the mosquito's ability to produce detoxification enzyme, and this has also been associated with its ability to adapt to these breeding sites.